Started.
Okay, great. Well, thanks everyone. Hello I think I know everyone, but I'm Jason Blackburn, a professor in geography with a pretty heavy dose of medical geography background. And today I'm really excited to introduce Dr. Michael Norris. Mike has been here at the university for several years, having first done a postdoc here, which is where many of us were introduced to him through the Department of Infectious Diseases and Immunology, which was over at the College of Vet Medicine as part of the Emerging Pathogens Institute, where Mike is currently jointly appointed in the emerging pathogens in a way similar to the way that Sadie, Ryan, and I are appointed, and also is an assistant research professor here in medical geography. So Mike's been in that position since September of 2018. And I think like many of our faculty and a number of students, geography was a found discipline for Mike, and he fits very well into our multidisciplinary medical geography program. So Mike's training was actually at University of Hawaii. He works on a number of environmentally maintained pathogens, which is where my collaboration with Mike started, which is trying to understand these pathogens that persist in the environment. And Mike's going to talk all about that today. So in that sense, Mike's work has already been sort of implicitly geography, and over the last couple of years here in the department has been more explicitly geography. So he's found the discipline, like many of our medical geography faculty, and contributes to that medical geography with a new perspective. And in this case, Mike's perspective as one who comes at it from the perspective of the pathogen out to the environment, rather than from the environment to where that pathogen persists. And he's had experience working on both ends, both working in the field at the landscape level, looking at outbreaks. He'll talk about some of those today. And then also where Mike spends a lot of time is in the pathogen lab, working directly with our pathogens. He's going to talk to you today about the fact that not only is he living through the pandemic with us, but he's also now a virologist as well as a bacteriologist, having set up a COVID lab here on campus and part of the university's response to the COVID pandemic. So he's going to be talking about both bacteria and viruses today, but also coming at it with a perspective of a laboratorian. So Mike sort of starts from the bench and looks outward. And so that's the perspective I want you to be thinking about as you sort of hear Mike's take on geography today. The other couple of things I'll point out just in terms of Mike's productivity, is this is also part of a job talk for medical geography. Mike has close to 40 peer-reviewed publications out already, and several in review, including some relatively recent big papers, such as invited papers in PLOS Biology, looking at the persistence of Bacillus anthracis from some of the strains in the collection that Mike and I co-manage of the anthrax bacterium. He's also the PI of a recent grant that he was awarded from the Defense Threat Reduction Agency, which is about a $2.5 million 5-year grant, to take some of his knowledge about the evolution and ecology of Bacillus anthracis and look at how different strains may or may not evade available vaccines. So he's also interested in disease prevention, and is going to be talking about that both from sort of a global perspective, but then also from how do you develop a vaccine, which is where a lot of his previous pre-geography experience lies. He's also, through his work on COVID, involved in several patents. So he's got experience with the UF Patent Office, which I think is another interesting addition to the medical geography program, and is co-PI in a grant with me in Vietnam that'll kick off here in about 2 months, which is a $4.5 million 5-year project linking the academic, public health, animal health, and military sector in Vietnam for studying Burkholderia, one of the pathogens Mike's an expert on. So Mike, I'll turn it over to you from there, and thanks for your time today.
All right, thank you, Jason. Let me share the screen here. Is that working, guys? Okay, well, I'll just assume
Yes.
that. Okay,
Yep.
thank you. Thank you for that introduction, Jason. I can skip most of that information on this slide, but so I've titled this talk Adventures in Infectious Disease, a World Tour, just to kind of highlight a lot of the research that I've been doing over the past few years in the Department of Geography, and also out of the Emerging Pathogens Institute, where I help direct the SEER lab and its high containment laboratory facilities. So I've divided this talk into three sections. The first will focus on capacity building and serosurveillance for the tropical infectious disease melioidosis caused by the bacterium Burkholderia pseudomallei. I studied this pathogen for about 15 years. That's how I can say that, because it's a mouthful. But a lot of people haven't heard of that in the US. It's a rare pathogen here associated with importation or overseas visits, but is found in the hot, humid soil environments throughout the tropics. Then I'll focus on the spore-forming anthrax-causing pathogens that are found globally. And I want to emphasize that I say anthrax-causing pathogens because it's not just a single organism. Typically, we think of the organism Bacillus anthracis causing anthrax, but there are some other closely related bacteria that have evolved to cause anthrax. Within this spore-forming bacterium research, I'll talk about our outbreak work in Texas, where we've aimed to characterize pathogen persistence and infection dynamics, and how we've involved genome sequencing to identify local, regional, and global pathogen emergence. And then lastly, we get a lot of information about pathogen diversity, and we understand a lot about that, but we want to also know, does that pathogen diversity affect vaccine efficacy, not only in the veterinary field, but also in the human vaccine space. And lastly, I have a couple slides about our pandemic COVID-19 work. As Dr. Blackburn mentioned, before the pandemic, the only thing I did with viruses was catch them. But now, there's a lot of need for high containment expertise at the University of Florida. And so I was kind of roped in to help with some projects that have ended up being pretty productive. One of them being our CRISPR whole genome screening project, and then our therapeutic discovery and testing projects. These are a little more heavily molecular, but I think it would be good to share them with the department. All right, so first off on our stop is Vietnam, where we study melioidosis. So like I said, you may not have heard of this disease or the pathogen that causes it. It's been associated with some high profile incidences in the United States. There was a large environmental release in Tulane associated with research activities. In this past fall, there was actually 4 fatal cases in the US. The CDC linked those cases to an aromatherapy spray. I know this pathogen as being highly infectious when aerosolized. And so this ended up with 4 deaths. This aromatherapy spray was sold at Walmart. And they found that at some point during the manufacturing process, the botanical extracts were contaminated with Burkholderia. This here is a picture on the left side of Ninh Binh, Vietnam. It's a beautiful area in north central Vietnam, characterized by these limestone rock formations. In between the rock formations here, you can see there are these flooded rice fields. This is the ideal environment if you would like to catch melioidosis. The Burkholderia pseudomallei bacterium thrives in this sort of environment. And like I said, it's a rare pathogen, so a lot of things aren't really understood about it, notably its ecology as far as animals are concerned. We know that humans can walk in these fields and through cuts in the skin can become infected, but we don't know whether animals play a role in spreading the disease or can be sentinels for animal infections. And so that kind of set up our project in Vietnam through the Science and Technology Engagement Program, where I was a project scientist. That the main goal was to understand the exposure of swine in north central Vietnam to the pathogen Burkholderia pseudomallei. It being a rare pathogen, the kits to measure seroreactivity with the pathogen are not commercially available. So down at the lab in the Emerging Pathogens Institute, I isolate out the antigens that we use to coat these plates, package them up into kits, and then ship them to Vietnam. And then I went there twice to help train the collaborators at National Institute for Veterinary Research in running these kits. So they obtained roughly 800 samples from farmed swine, and then 300 to 400 samples from grazed swine that we could run these kits on. And so we did that. We ended up with a publication with them. This is the data from that publication. The main point being that down here in D, we can see that the y-axis is the reactivity with our assay. And we had two different assays, a capsular polysaccharide assay and another outer polysaccharide assay. And we see one of them is, we know, is more sensitive at detecting reactivity to the pathogen. When we break that out in E to the different communes within the two provinces we studied, our study site was in Ha Tinh, we had some reactivity in Thuong Loc commune. To the CPS test on the right-hand side here, we had high reactivity in Vu Quang and Song Kim and Phu Lu in Ha Tinh. And this was among farmed animals. So this is a sort of a denser, more industrial facility. What we noticed that was in the grazing animals, we had sort of across the board higher levels of seropositivity there. And we used all this information to inform one arm of our DITRA BTRP project that Jason mentioned, Dr. Blackburn mentioned before in the introduction. This is the biothreat reduction program. This project is going to start this spring. And so moving now from melioidosis in Vietnam, or actually, we also utilized this data to understand the number of animals at risk in these communes that we were studying in. In the middle here, we have a map of Vietnam. The upper panels are the seroprevalence we measured in the two different provinces and those communes within them. The darker the color, the higher the seropositivity rates. The darkest colors are around 20% seropositivity in our samples, and that's relatively high. Normally, we can find 6% in other animals, indicating potential hotspots of melioidosis in these provinces. Another interesting aspect that we're able to analyze is the number of swine that could potentially be exposed in our study areas. There is the Gridded Livestock of the World database, which provides livestock numbers for every 5 minutes of arc on the globe. And that's roughly an 8 square kilometer area. This is a really neat database. You have numbers for ducks, for cattle, for horses, for sheep, and also swine. And so we can estimate in our areas of a high seroprevalence roughly how many swine are seropositive to melioidosis at any given time. And so we have, between 600 and 700 swine in our hotspots at any given time that could be seropositive for melioidosis. The same is true in our Hoa Binh Hoa Binh province in our grazed animals. Lastly, what we did was we plotted our seroprevalence across the communes within our two provinces. Again, the green are farmed animals and the red are grazed. And this sort of highlights the fact that I mentioned earlier that a couple farming operations had high levels of seropositivity while others did not. And this is likely indicative of the shared food and water resources within those operations and the known possibility of pseudomelite contamination of those water sources. The free grazing animals all seem to have elevated seropositivity rates. And we think the grazing animals might be a better sentinel animal for understanding human exposure to Burkholderia pseudomallei in the environment. So that kind of wraps up the Vietnam study. We've started another similar study with collaborators at Cornell that's funded by the Atkinson Center for Sustainability, where we're looking at swine seroprevalence to melioidosis in Uganda. And so that's kind of ongoing. We just got some good results from that. And then we're also developing a project similar to our Vietnam project in Malaysia with government and academic partners there. And we're in the pre-proposal stage for that. So now we'll go from melioidosis in Vietnam to anthrax in Texas. So the Seer Lab has had a long-standing collaboration with some ranches out in Texas in what's called the Anthrax Triangle. And we got a call in 2019. It happens a lot actually around here. You get a call that there's an outbreak or there's an infectious disease incident that needs to be researched. In this case, a veterinarian called and said, in the summer of 2019, this is the largest anthrax outbreak in my lifetime. And these guys have been around for a while. And so we packed up and flew over there and then drove out to this area, which is near the border of Texas and Mexico in West Texas. A very interesting landscape, a lot of sort of semi-arid, not quite mountainous, a lot of limestone rock, and then seasonal rivers. And they've got all these ranches. Some of them are very large. Most of them are pay-for-hunt operations. This picture of the giraffes is not a pay-for-hunt. This is a conservation ranch, but it's a rare occurrence out there. A lot of very exotic African mammals out there. I can't remember them all. We've got kudu, gemsbok, springbok, axis, impala, aoudad, just a huge diversity of mammals out there on very large ranches up to 20,000 acres. Some of those ranches are exotic animals. Some of them are deer specifically. But while we were out there, me, Dr. Blackburn, Morgan Walker, and Ted Hadfield, we sampled deer carcasses in all over the place. We took GPS coordinates, and we took environmental samples for analysis back at the lab. That's me. You probably may have seen a picture of me around the office. It seems like it's all I'm doing is wearing Tyvek and a protective breathing PPE. But in this case, it's very necessary because the animals, as they're dying from anthrax in 110-degree heat, they try to get into some shade, and they end up creating a very contaminated area that we got to protect ourselves against. In the upper right here, this is a typical deer carcass that's maybe a week or two old. You can see the area around it is stained with the fluids of that dead animal. Those fluids are highly contaminated with anthrax spores, so that's Bacillus anthracis. This can create contaminated areas, for years or decades to come, that we call a localized infectious zone. So it's not all fun and games out there. We also help the ranchers clear the land of their dead carcasses to try and help reduce risk for anthrax on their properties later. And that includes scooping them up in these bulldozers or loading them onto trailers. It's kind of a messy business, but in the end, you end up decontaminating those carcasses and killing the spores with by burning them at high temperature. So we're out there collecting samples to answer some of our hypotheses about what's causing these outbreaks. So we know that usually it occurs in the summer after a wet spring, and then there's also usually an abnormal rain event in the middle of a summer, the summer heat. And then anthrax outbreaks kind of start after that. But what is perpetuating these anthrax outbreaks is probably another mechanical aspect, and we think it has to do with flies. So there are two types of flies we're interested in out there, and that's necrophagous flies, which can be seen here in the bottom picture. This is the surface of a dead deer. You can see the flies on there. You can also see these little black specks, and that is fly emesis. So the flies eat the anthrax contaminated carcass and then defecate all over everything around it, basically. Here's a picture of a rock covered in those little speckles, dappled in it. That's all contaminated with anthrax spores. I don't have a picture here, but I mentioned, the animals search out shade. And so if you go into an area where there's a carcass and there's some trees above, you can see there's like maybe 1,000 or thousands of these necrophagous flies, and it's just glistening with anthrax contaminated emesis. So we've got to be very careful, and all those surfaces could create potential contaminated surfaces that other animals, if they're curious about the food or the carcass, can then be infected and carry on the outbreak. So another aspect that we're interested out there is vector incrimination. So up on the upper right here, you see this is a picture of a biting fly on the surface of a dead axis deer. And so this particular animal, we were doing a transect, so we weren't trying to isolate any samples, but we came across this animal that died in front of us of anthrax. And then these flies usually feed on living animals, but as long as they're warm, and so they'll feed after death. And so we observed this dead animal, and the flies started taking their blood meal from the dead animal. And then us, being industrious scientists, captured these biting flies in Gatorade bottles. That's all we had on us at the time. And then we took a sample from the turbinates of the deer, and then took that back to the lab here at UF. The lab team then, including myself, we ground up the fly, and we found about 100,000 anthrax spores in that fly. That's, for perspective, that's enough to kill maybe 50 to 100 guinea pigs, or any number of deer. And we also isolated the bacterium from there and sequenced the genome of the fly, the bacteria that was found in the fly, and the bacterium that was found in the deer. And when we align those genomes, they're 100% accurate. And so we think this shows at least part of the vector incrimination, that the fly can, pick up enough infectious anthrax spores to cause a lethal infection down the line. And that's sort of the next step we're trying to answer is, can these biting flies deliver an infectious dose during a bite of an animal? And so that is where we are moving towards next. We're also interested in the persistence of these wild strains, we call them. These are strains that are directly from outbreaks. And one thing we did was compare them to laboratory strains. So laboratory strains are the strains that scientists use. Generally, they grow them a lot to understand the physiology of the organism in model systems here. And when we analyzed the proteome of the vegetatively growing Bacillus anthracis, we're able to separate the wild strains from these laboratory strains in this principal component analysis of their proteome. An interesting finding when we did the proteomics analysis was we found a lot of proteins involved in sporulation of the bacterium and growth of the bacterium, particularly in the wild strains. And then through laboratory experiments, we showed that the bacteria grow vegetatively. So these red dots are higher than the blue ones. They grow faster, and they also sporulate faster than their laboratory high passage counterparts. And we think this can help inform public health knowledge out there in Texas, because a lot of times the ranchers, they know the animals die, but they don't do anything about it. They're either scared or they just don't care. In this case, we think that, as soon as they know an animal's dead, they need to either dispose of it through burning or other means of decontamination. Otherwise, they end up with this sort of thing where a year later you've got, a localized infectious zone that is, you could probably still isolate anthrax spores from. Now moving from anthrax in Texas back here to EPI. We've been developing a pipeline for pathogen genome sequencing. So we work with some dangerous pathogens. We can't just isolate the DNA and then ship it to somebody to sequence it. There's a little paperwork that goes along with that. And so to avoid that, we established this in-house field to genome sequencing pipeline. We also do the bioinformatics analysis in-house and on the Hypergator. This is a picture of the MiSeq Illumina machine that we use for next generation sequencing at the EPI. We get around 15 billion base pairs if everything is optimized when we run a cartridge there. And thus far, we've sequenced about 48 genomes of Bacillus anthracis from the Texas outbreaks, around 100 genomes over the last 2 years total for various projects. And I had to mention this example because, again, we got a phone call from the Gainesville VA because we have a reputation for being able to figure these things out. That said, hey, we have a potential brucellosis case, but the state lab can't confirm. And so our laboratorians isolate out the bacterium, we sequence it, put the genome together, and we help them identify it as a Brucella suis infection in an individual at the Gainesville VA. This is another interesting story. Again, another phone call. The UF Veterinary Medical Campus, they had a kangaroo from Sumter County, Florida, which is down just past Ocala. They had a kangaroo that died. This was some goat yoga thing, and I'm not too clear on why there were kangaroos there, but either way, the animal was sick and they had to euthanize it. And what you can see in this image, this is the histopathology of the tissue. And they found a bunch of vegetative bacteria. These are just normally growing bacteria, which are these sort of dark colors here. And then they found sporulating bacteria, which it's hard to tell, but spore is like the dormant form of bacillus or clostridium. In this case, the veterinarians thought it was Clostridium botulinum, which causes botulism. Initial tests showed that it was a bacillus species, but they couldn't confirm what type exactly. And so then, again, we had a laboratory team mate. Everything okay? Okay. We had a laboratory team was able to isolate the bacteria from this tissue, and then we sequenced it in-house. We looked up the sequence types from the whole genome sequence, and we identified 2 other strains of Bacillus cereus. So as I mentioned before, I said anthrax causing pathogens, not Bacillus anthracis, because there are other bacteria such as this Bacillus cereus, which are normally apathogenic. But what's happened in the environment is they take up genetic elements from Bacillus anthracis somewhere along the way that allow them to secrete toxins and create capsules that make them anthrax causing bacteria. And interestingly, this G9241 that showed the same sequence type as our kangaroo strain was found in a pipe welder's infection in Texas. And it was exactly similar to that. And so it was a Bacillus cereus in the chromosome, but it also contained this. This is a whole genome. These are genome sequence alignments I'm showing you that show that it had the toxin encoding genetic element associated with anthrax. And what else popped out to us was this BC Florida 2013 strain that shared the sequence types with the one from Texas as well as our roux. And we looked at this paper and found that it was isolated from a person, what we've been told is down near Ocala. You can tell this is someone's face. You see there's an ear there. This is what we call an eschar. An eschar is this typical cutaneous manifestation of anthrax. They isolated the pathogen. I don't know if I'd let it get that bad before seeking help, but this is an eschar caused by Bacillus cereus. And it turns out our kangaroo died from a similar strain from the same area 8 years after this human case. And so what we think this indicates is the establishment of an exotic pathogen down there in the environment, whether it's in the soil or being spread by insects under the right conditions, we don't know, but I think there's going to be some more interesting findings there. So from anthrax in Texas, anthrax in Florida, let's take a visit to West Africa, West and Central Africa. So this is another interesting story. Typically, we think of anthrax occurring on in these sort of open savanna, semi-arid areas full of herds of mammals, ungulates. But what was happening here in West and Central Africa about 20 years ago was there was a lot of anthrax going on in the rainforest, which isn't really typical of anthrax itself, of Bacillus anthracis anthrax. And more confusing was that it was occurring amongst primates. And a few years later, they figured out that it was not Bacillus anthracis causing these anthrax outbreaks in the red areas amongst primates in the rainforest, but it was another Bacillus cereus that had obtained those toxin and capsule encoding elements. They named it then Bacillus cereus biovar anthracis, or BCBVA. Also occurring in especially Thai National Park, it was an intensive research outpost for the Koch Institute out of Germany. It was for a long time an outpost where they were studying primate behaviors, primate social organizations. And so they're anthropologists interested in the animals living in this forest, as far as understanding. They take the bones, they would measure the bones to understand the health of the animals. They would saw the bones in half to understand nutrient content of the animals. And they would send these bones to anthropologists around the world that were interested in this subject. Some of those anthropologists and those bones made it to the building that I would be in if I didn't have Omicron right now, and that'd be Turlington Hall, in the basement there. And again, somebody from the Thai National Park that realized they had an anthrax problem called the anthropologist they had sent all the bones to and said, hold off on sawing those bones open. They might be full of anthrax. And so obviously our collaborators didn't want to be involved with any accidental anthrax cases. And so the university tapped the SEER lab to take these bones and analyze them in our high containment facility. It turns out some of the bones contained Bacillus cereus biovar anthracis spores. This is the first one that the lab I was able to isolate spores from. This is a tooth. You can see there's tissue around that tooth. Bacillus cereus biovar anthracis spores were isolated from that. A few other bones dating back to 1994 also had spores in them. And so 1994 is roughly 10 years before they even realized, before scientists realized there was an anthrax problem in the forest amongst primates. And so that strain we have is like the earliest ancestral snapshot of Bacillus cereus biovar anthracis that's around. So obviously, having BCBVA and Bacillus anthracis causing anthrax in the same area can be confusing to diagnostic laboratories. And so we're working on helping those folks there understand how to correctly diagnose anthrax caused by Bacillus anthracis or BCBVA. And we also interested in the genetic signatures from these pathogens, so that we can understand the spatial epidemiology in the rainforest. So what I'm showing you here is phylogenetic trees of whole genome SNP alignments of all the BCBVA sequences available. Each one of these little labels is a different strain. And if there's no branching, which you see here, there's no branching, looks like a pinwheel, that means that the genetic similarity, they're identical. And this, in the upper left, this is the toxin encoding genetic element alignment. This is the capsule encoding genetic alignment. And we don't really see any patterns there, right? You don't see any light colors near each other. It looks kind of random, and there's not much branching amongst the branches here. And so we basically discount those as having little diagnostic value for our purposes. But down here in the chromosome, we can see there are patterns that start to emerge. We can see that these branch labels as that color is warmer, it means it's an earlier isolate. The blue is all from 2014, so there's a lot of isolates from 2014, going all the way back to red, which is 1991. And we can see there are sort of these three organizations of temporal pattern here. On the outer ring here, we have country of origin. So a lot of these samples are from Côte d'Ivoire. We have a few from Liberia, one from DRC, one from Cameroon, and a couple from the Central African Republic. But those all cluster together in this region, indicating what we think is sort of a regional outbreak of this homogeneous pathogen. So now we have all this information, we wanted to know, can we use this genetic information to understand spatial patterns at the local level? And so we had about 55 of these strains that we had GPS coordinates for, and we looked at their geographic distance and tried to understand patterns in their genetic distance. So here we did a weighted Ripley's K test to try and understand whether if strains were geographically close, were they genetically similar? Sort of a test of the geographic spatial decay theory. And so what we saw is that, and this is a very small area, I wanted to mention that first. So Thai Park is rather large, but this study area is, maybe 10 kilometers. All of our strains are within probably 2 kilometers of each other. And so we're testing this at very small spatial scales, at least for most of the work we do. And we see here from around 350 to 900 or so meters from one strain to another. We see more genetically similar strains clustering around them than can be explained by random distributions. And the same is true when we look at our two earliest strains here. The later strain here on the bottom right, it still falls outside of the confidence interval, meaning there is a significance in that similarity and distribution. And that's something we're continuing to look at. Another way we can visualize this is with kernel density estimations. Again, like I said, this is a very small, maybe 2 to 4 kilometer area. But we can see some spatial patterns. Each dot represents a carcass where a strain was isolated from that we have GPS coordinates for. And so we found it interesting that we could even identify patterns at such a small local level. And I think it's going to be interesting when we apply this to larger study areas like we have in Texas. All right, moving from BCBVA in Africa to back to the lab here. I'm going to talk about that project that we got funded in September involving whether or not current next generation vaccines are effective against diverse anthrax strains. So this project, I serve as PI on here at UF. We're working with Colorado State University in the guinea pig challenge model to measure vaccine efficacy. And we're also working with the US Army that will provide their next generation vaccines for testing. The whole question with this project centers around whether or not vaccines generate antibodies to protein and non-protein components of the spore. This is what we call a Western blot. The Western blot, we can have whatever material we want in these lanes, and then we take immune serum. This is serum from humans vaccinated with the human AVA vaccine. This is a purified subunit vaccine. On the right-hand side here, we have bison serum from bison vaccinated with a veterinary vaccine. This is a whole spore attenuated vaccine. So it's a live spore. And this is one they use all over the world. They use it for livestock, wildlife, you name it. And so what we've run on these blots are vegetative Bacillus anthracis in lanes 1 and Bacillus anthracis where we've gotten rid of all the proteins. So all there are sugars that remain there. And so we can see that it doesn't matter which vaccine, we only have reactivity to proteins of the vegetative cell. In lane 3 and 4, we have we've run Bacillus anthracis spores, total spores in lane 3 of each, high reactivity. And what we see that's different than the vegetative bacteria is that we have, after we remove all the protein, we have reactivity to a sugar component very strongly in the veterinary vaccine, which makes sense because it's a whole spore vaccine. And so you get antibodies to the spore in that vaccine. What we weren't expecting was to see a little bit of reactivity in the human AVA vaccine. So this vaccine technically doesn't contain any spores or material from that, but we still see reactivity with a sugar component in the spore. So I hope that makes sense, and keep that in mind for the next couple slides. We're also going to, so Tamburini et al. In 2011 found that certain strains from West Africa, from Mali, Chad, and Cameroon contained genetic deletions that resulted in removal of a key sugar from the surface of the spore. In these same strains, Blackburn et al. Found they were genetically distinct amongst Nigeria, Chad, and Cameroonian strains in what we call the WAG Bacillus anthracis. So I know what is the sugar? Well, it's called Anthros. And so those strains that have mutations in there don't produce Anthros on the surface of the spore. This is what a spore looks like. We made and took this picture ourselves here at UF. You can see the sort of fuzzy area on the outside of that spore. That's the exosporium nap. It's kind of like this Velcro area. This is the very outside of the spore that interacts with the environment. While it's infecting a host, it's that first barrier there. And on that surface, you have these protein stalks that are decorated by sugars. And at the very end of that is Anthros. And so what's happening in these bacteria is that they are mutating in their DNA so they don't present Anthros on the surface of the spore. So that could have some far-reaching effects on vaccine efficacy and pathogenicity and environmental persistence. And these are things we're trying to answer. But along the way, we're just trying to grasp how big of a problem this could potentially be. I have on this map here the WAG strains. And during our studies, we found additional strains through totally different genetic mechanisms becoming Anthros negative. We found a strain in Chile, strain in Poland, and this one's really interesting. We're calling this the heroin anthrax. So what happened here is this strain was associated with an anthrax outbreak amongst heroin users in Scandinavia and the UK. Somehow, anthrax spores got into a bunch of heroin, and when it's injected, it's highly virulent and killed several, I think about 80 heroin addicts. And what we're showing here in this phylogenetic tree is that even though they're genetically and geographically very different, they still arise at Anthros negativity via their distinct mechanisms. This is just, we've done some modeling where we ask, is there an external pressure pushing strains toward Anthros negativity? And at this point, we have to say yes, there is. So that was one paper we published in PLOS Bio. Since then, we've identified some other mechanisms and new strains from other places, such as China. We also have this djembe drum related anthrax outbreak. And so what happened here is an animal died of anthrax. That animal skin was then exported to make djembe drums. And in that process, they had anthrax outbreaks in the USA, UK, via Senegal and Gambia is the assumption there. And so besides this heroin strain, the one I talked about in the previous slide, there's actually a whole bunch of heroin strains. So I'm not sure, I don't think all of them were found in heroin, but they're called the heroin anthrax strains now. And these are all different mechanisms of Anthros negativity. So we first started trying to understand this with the West Africa group anthrax, but now it's turned into a larger issue, we think. And one we're also trying to address in the Philippines with our STEP project there. All right, so one more phylogenetic tree, I think. This is just to help illustrate that diversity of strains that are Anthros negative. So remember that Anthros negativity changes the surface of the spore, and whether or not that affects vaccine efficacy in animals is something that we're in the process of determining. But you can see each branch that is red here is a different Anthros negative strain. And each of these are from very different groups. So the West Africa group, South America group, this one's from China, the drum clade from West Africa, and the heroin group from Turkey. And these heroin strains have been found in the US back to 1960. And so we're in the process of analyzing these strains. First of all, what does Anthros do besides have a potential impact on vaccine efficacy? Well, we knock out Anthros production and we generate spores. And we looked at, we digitally peeled the exosporium off, created these 3D topographical surface plots, and found that removing Anthros decreases the density of that exosporium nap. And we think, and we've shown before that removing Anthros can increase interactions with immune cells. And so we're also in the process of getting this published in PLOS Bio. We've also been analyzing the toxin secretion. And so this is more on the pathogenicity side. So when we remove Anthros, we get higher levels of lethal factor secretion. This is the toxin secretion early on that goes away later on. And so we think that's why, and this is the Galleria melanella model. So we generate spores and we inject them into the larva. And we can see the red line here is our Anthros negative strain that kills those larvae faster. And so we're in the process of doing these same studies, but with guinea pigs at Colorado State University. All right, so I hope that's clear about the Anthros. If it's not, we can talk afterwards. But moving on to SARS, because it's sort of impacting us all still. It's been a productive collaboration to do the SARS work. Probably been spending more time doing it than I should. But we call this jovially the UF COVID CRISPR Consortium. It sounds official, but it is not. And this collaboration was started because a lot of people had a lot of ideas to get work going, but just weren't able to do BSL-3 high containment work. And that's where my skill set and our team's skill set came in. This project involves generating whole genome CRISPR knockouts. This is, bear with me, it's very molecular intensive. And then we take the whole genome screens, infect the virus. Whatever comes out is something that survives the infection. And that means they have a genetic knockout in something the virus needs to replicate in our cells. And then those host factors that we identify are great targets for drugs or for gene therapy. We have done a lot of this work. This is just kind of to illustrate what we get out of it. We get a lot of genes up here that are important. We've done a lot of screens, and we've come up with a couple mechanisms we really want to study. We knock out these genes in human lung cells, and we can see that when we knock those genes out, we reduce the growth of the virus of SARS-CoV-2 or the common cold virus in those cells. We've also identified 6 inhibitors of various genes we identify there. Inhibitors meaning small molecule drugs in SARS or OC43, which causes a common cold. We have some preliminary data showing that one of our inhibitors, Amlexanox, can reduce viral load in the lungs of mice. And also when we use gene therapy, so we actually do gene suppression with the treatment in mice lungs, we can reduce the viral load there. This has been we have a couple internal funding projects here. We completed a CTSI project. We have Office of Research money. We have Research Opportunity Fund money. We've got 2 patents. And finally, we've got this paper that's accepted in genome medicine. So that should come out soon. There's more about this in the fall 2021 UF Explore magazine on page 6, if you want to read more about that. Lastly, I'll talk about our inhibitor research. This is a figure we made for our paper. This is the life cycle of the virus in the cell. And this collaboration kind of started initially between myself, Leon Resnikoff, and Dr. David Ostrov. they found that antihistamines, some of them common ones, inhibited binding of the virus's spike protein to the ACE2 receptor, which is the receptor on the surface of our cells that is required for the virus to infect us. In their model, they weren't using live virus. And so then in our lab, we ran some of these assays where we just tested, do these antihistamines prevent viral replication? And yes, some of them do. Hydroxyzine is here. This is diphenhydramine, more commonly known as Benadryl. And then the last one here is azelastine, which is like a nasal spray antihistamine. Oops. So that kind of led us to another aspect of the viral replication cycle, is down in here in this area where the virus is doing its thing. They were thought to interact with sigma receptors, which are also impacted by antihistamines. And sigma receptors are interesting in that they are involved in pain reception and also addiction. And it's so happened that we have a sigma receptor specialist here in the Department of Medicinal Chemistry, Dr. Chris McCurdy. And Dr. Ostrov and I reached out to him, and he had these compounds that he was using in studying drug addiction and pain. And it turns out these very specific compounds actually inhibit viral replication in human respiratory cells. And that was a very interesting finding. And we got a little money from the College of Pharmacy, their Prosper seed fund, to test this in mice. And that's going on right now. I think Andrew Bloom is on the phone from the lab doing that right now. Good job. And then the far right here, we also looked at the combination of Benadryl and another drug that we're also interested in studying. So by all means, don't go out and buy Benadryl unless you really want to. But I've been taking it. So with that, thank you. I want to thank some folks involved in the anthrax amyloidosis work, especially Dr. Jason Blackburn, the SEER Lab founder, SEER Lab members, Ted Trinate Morgan, Matraylor Morgan, Hawk Walker, newest additions, Dr. Jamie Ben, Dr. Sam Sawyer, undergrads, Madison Burns, and McCantac. The COVID work, the consortium is Dr. Karsh, Dr. Volpe, their team. We've been working with David Bloom and his grad student, and of course, Dr. Ostrov and Chris McCurdy. And I got to mention Andrew Bloom. He's a skilled high containment lab scientist who, somebody gets COVID, say, he continues that work in the lab unabated. And lastly, our funding agencies, the DITRA, the internal funders, and especially the UF EPI director, Dr. Glenn Morris. He's been supportive of all this work over the years. Thank you.
Thank you very much, Mike. You've got some rounds of applause. Now you've turned it off, I can see the little things. So does anybody have any questions for Dr. Norris related to his presentation or general? I have to keep going back and forth on this screen to see everyone. Okay, I'm going to start off. So if anyone has questions, kick in and let me know, but I have a few, some of which may be a little bizarre because, as Morgan will tell you, I'm very into sequencing. Mm-hmm. Okay, so one obvious question for the switch in position that we're looking at is making it so that we can take you out of the lab sometimes and out of the field and put you in a classroom. Yeah. And the presentation you gave is great in terms of showing that it will present to us the full suite, right? We can go from actually obtaining whatever the disease is, through kind of sequencing and biology of it, through spatial analysis, and kind of taking it up to the what if and what can we do. So it's kind of end to end, which I think is pretty novel in a MedGeo program. But in terms of thinking of what is already part of the MedGeo program that we have here at UF, what do you think in terms of courses that you would like to offer and teach, given that we would be able to get you in a classroom with this switch? And how would they kind of fit in terms of both your own research program and kind of graduate training?
So I think a good class that would be very beneficial to the program would be even like an introductory laboratory skills class, where, we're not talking intense biology courses, but just enough to know, well, this is how we get that answer, right? If we need to design a new test, which happens a lot, this is what that means biologically, right? So I think an introductory wet lab course where we can get students into the lab doing some basic molecular techniques would be really worthwhile. And then building on that, a more complicated sequence-based analysis course where we can start to tie together some of these genetic findings, like we're doing now with, we use what's called multityping to understand diversity of anthrax, and then tying that back into spatial understanding, especially on the Texas landscape. We're doing it in Africa at local scales, and also Morgan Walker's doing it at global scales. So some folks in the lab are already doing these sort of things, but really, I guess standardizing it in a class is what I would be interested in doing.
Okay, so given a number of my meetings today have been on AI, both curriculum side and research side, and you didn't mention AI at all, but when I'm looking at your research, and I think from reading some of your papers, which we've done in Geographic Thought, it seems like you're definitely in that realm of big data, machine learning, hypergator, and AI. Are you?
Yeah. I mean, the amount of data we get out of a sequencing run, 15 gigabases, that's 15 billion base pairs. That's just one run. And when we analyze that data, it does take using hypergator. When we try to analyze patterns in that, that's definitely we get into the AI realm of things, machine learning. And so I'm definitely interested in expanding on that with some of our global questions, right? So when these data sets get that big, you need some high computing environments to do that. Yeah.
I mean, obviously for UF, love it or loathe it. UF is heavily invested in AI, and that's not going away before 2030, given current investments that are being made. So it would be nice to have another kind of node in geography linked directly with AI.
Sure.
So one completely off the top, and I don't know if Brian is still on here, he was on earlier, but I'm sitting there and suddenly giraffes appeared on screen, which surprised me. I did not know we had giraffes in Texas. But then you mentioned that those are kind of working, well, that one you said was a conservation landscape, but that a lot of them with the kudu and everything else are these functioning hunting landscapes. And Brian studies the exact same landscape in Africa, and he actually works, there's Brian, he actually works with a lot of those groups, and he's obviously looking at kind of the monetary side and looking at, how they can work for local communities so that they're getting that income. But it just seemed like you described a landscape that Brian is studying in Africa with similar wildlife and systems. And it just seemed like there was potential synergy in those. And I know you work in Africa somewhat. And I know, I think, Jason, you had a project in Chobe at one point, I think, with anthrax. So it was just kind of interesting that I'd never really thought of Brian's conservation landscapes and kind of the MedGeo work that the CO Lab does as even having a connection. So that was kind of an interesting potential synergy there. Not really a question, but it just.
Note taken. I agree. I mean, I've not been to Africa. I've seen pictures of it, but, I was out there in Texas and I could have, pretended I was there.
We'll show you some kudu.
Yeah, live ones, preferably.
Yeah, you got to learn the names in the first 2 days.
Right.
Are there any other questions from anyone?
Jane.
Yep, Vincent?
Yeah, on that note, that picture and the one that you mentioned about conservation oriented, was that Burr Ranch by any chance?
Was that what?
Burr Ranch?
I can't comment on any specific ranches there, but it was definitely a conservation ranch.
Understood. And then as far as with the impacts from those flies, you guys cited a lot of the deer species. Did you notice that in those larger host stock species, giraffe, etc.?
The flies themselves?
Correct.
Well, so most of the, the animals exhibit behavior that shoos the fly away for the most part. And we didn't see any, actually, none of the first thing they did was vaccinate the giraffes when they heard there was an anthrax outbreak. So those animals were protected. So we didn't see any dead giraffes, thankfully. But we didn't really observe any biting flies on the giraffes. They were very healthy and kind of shooing them away. They have that long tail. Yeah.
Any other questions? I think we went a little bit long. It's 4:01, but thank you everyone for hanging in there. And thank you, Dr. Norris, for a very interesting talk. And next week it's me, so it's not that interesting in many ways. It is very much about process and about teaching repositories and teaching archives that we're trying to develop both for faculty and especially for the grad students. So we will have that meeting next week, which will be a little bit interactive as well. So hopefully we will be in person. Hopefully I will be allowed back. And hopefully Mike will actually recover and be fine. And, thank you for giving a talk even when you probably feel like death.
Yeah, no problem. My pleasure.
Thanks, everyone. Have a good weekend.
Bye.
Thank you.